This next section, we are going to continue on our discussion of tissue, but we're going to move into muscle tissue, which is our third tissue type. Thus far, we've looked at connective tissue and epithelial tissue, and muscle tissue has three different types, which include skeletal muscle, smooth muscle, and cardiac muscle. We're going to identify that with our different muscle types, we will see that our skeletal muscle type, which is shown on this slide set here, essentially in histological view, is striated. It has these striations or stripes from our sarcomeres. It contracts via voluntary action, meaning we decide when we contract our skeletal muscles, and it's attached to bone. Our cardiac muscle also contains striations, although they're a little bit lighter in hue than our skeletal muscle. This contracts constantly. It's our heart and it's involuntary. I don't have to tell my heart to contract, although I can influence my heart muscle through things like inducing my parasympathetic nervous system. Slowing down my breath, doing deep belly breathing is going to introduce or encourage a parasympathetic state, which can slow my heart rate, for instance, but it's involuntary. I'm not making a conscious choice to have my heart contract. And then smooth muscle. Smooth muscle's non-striated. There's none of those stripes that we see in the other two types. It also contracts as non-involuntary. And we see this in our soft tissues, some organs, and our vessels. What are some features then of skeletal muscle? Kind of going back to the top, we'll use skeletal muscle as kind of our primary muscle moniker, and then we'll compare and contrast to the other types of muscles. So we have muscle fibers, which muscle fibers are essentially a muscle cell. And these are multinucleated, meaning they have more than one nuclei, and they contract as a syncytium, meaning that they contract kind of all as one. They are these long tubular cells, and they span the entire muscle length. When they contract, it's voluntary and it's via these myofilaments, which are these fibers inside an individual muscle cell. And these fibers are organized, as we can see here, in sarcomeres. And it's from this essentially green line to green line. And these sarcomeres are what make our striations on muscle. Now, thinking about muscle as an organ, muscle has connective tissue coverings. On the outermost portion of the whole muscle, like we could think of my bicep muscle, for instance, the whole muscle belly itself, as I was saying, our muscle itself is covered in connective tissue, and one of the connective tissue coverings is the epimysium. This covers the outer portion of the whole muscle itself and its dense, irregular connective tissue. Now, inside a muscle, though, as an organ, there are multiple different kind of compartments or um, smaller segments of the muscle, and these are called fascicles. Another way you could think of a fascicle is you have multiple muscle cells that are clustered together in a fascicle. And a fascicle itself is covered by perimysium, a connective tissue layering, which is also dense, irregular connective tissue. And then our smallest individual muscle fiber, or myofiber, is the same as a muscle cell, and it's covered with loose connective tissue that is endomysium. So you could say that a muscle itself is made up of multiple fascicles, and a fascicle is made up of multiple muscle cells. A muscle is covered with epimysium, a fascicle is covered with perimysium, and a muscle cell is covered with endomysium. These are all connective tissue. Now, zooming into an individual muscle cell, this individual muscle cell contains multiple myofibrils, and these myofibrils are made up of fiber. And these fibers can be called myofilaments. One myofilament is going to be actin, represented here as this green filament. This is the thin filament. The other filament is myosin, and this is the thick filament. When we look at our sarcomere, we have lots of different structures that make up kind of this overall contractile component of muscle, and we can label them as such. In the center of this structure, we are going to have our M line, and so our M line is going to be here. It's also going to be here at the center of the structure. The M line is what really anchors our thick filaments in place. We then have another line. This is called the Z line. You can't really see the Z line on this side here. You can see it over here. This is the Z line here. We can really see it well in this image. So this would be our Z line on either side. This is what anchors our thin filaments or anchors our actin. So those are our two lines that we have here and here. We then have two bands. We have an A band and an I band. The A band is going to be this center portion that contains our actin and myosin crossover. And it's going to be on either side of the M line. That's going to be our A band. Our I band is actually going to be consisting of just the thin filaments, just actin, and it's going to overlap our Z line. So the I band is here to here, here to here, and the I band actually is going to encompass more than one sarcomere. And then outside of our bands, we have one zone. The zone in the center, the H zone, is this central portion that goes around the M line that just consists of myosin, thick filaments. So the M zone would be here, this central portion here that's lighter. And it's within the greater A band, essentially. Now, when we think about how do muscles contract or sarcomere contraction, we'll see that our I bands and our H zones will shorten. So our I are essentially I band here and here and here. That's half of our I band. Gets smaller when we contract, right? It's much thinner here and here. And then the H zone, which was from here to here, is much smaller now. It shortens. We can see that this central A band, though, does not change shape. It stays the same. This shortening, though, of our I band and our H zone that represents muscle contraction is from myosin heads walking along actin filaments when muscles do contract. And these striations that we see histologically are from our sarcomeres. And this is a longitudinal section of muscle, skeletal muscle. We can really see those striations versus stippling, which kind of almost looks like marbling, is caused by myofibrils, those long myofibrils that contain multiple sarcomeres. And this is due to a cross section of skeletal muscle. A pathologic connection of skeletal muscle contraction is Duchenne's muscular dystrophy. In the histopathologic slide, we can see that there are multiple degenerating fibers where they were used to be pink, they're now almost white or clear. And this is due to increased connective tissue that's been laid down between muscle fibers and then has been replaced by adipocytes or fat cells. So if you're like, this looks like fat cells, you are correct. The condition results from an inheritance of an abnormal dystrophin gene, and dystrophin, as we'll learn in physiology, is a sarcolemma membrane stabilizing protein. Therefore, when it's gone, our plasma membrane is less stable in these muscle tissues in these individuals. Transitioning to cardiac muscle fibers are chains of branching mononucleated cells as one fiber. So these cells are short and narrow, and the cells in a fiber adhere and they communicate via lots of cell junctions that are located at these things called intercalated discs. These intercalated discs allow for this really good communication between one cell to the next to allow for a syncytium or a singular contraction at one time. The muscle fibers themselves are surrounded by endomysium, and they are post-mitotic. They're no longer dividing, undergoing cellular division. They undergo individual or involuntary contraction via myofilament shortening that's also organized into sarcomeres, and we can see the striations. So again, we can see these muscle cells here branch off. We have branching components branching by both myofibrils intracellularly, and we have these connections between cells via our intercalated discs. And there is a central nucleus in these cells and in these branching cardiomyocytes. This is what an intercalated disc looks like under an electron microscopy, so zoomed really, really far in. And we can still see these striations here, these stripes. You can see the striations here and the stripes. They are caused by the sarcomeres. They do have striations, and we can kind of see these intercalated discs listed or held in between cells. There is still also stippling. It's even more aggressive in this image caused by the myofibrils. In an acute heart attack or acute myocardial infarction, there is an abnormal striation pattern, as we can see in our muscle fibers, and there's also an absence of nuclei. We've lost our purple. This is because these cells are dying. They're unable to replace or regenerate with a healthy muscle cell. Therefore, they replace via non-contracting, non-conductive connective tissue. And that means that the flow of electrical impulses to the heart can be disrupted because we no longer have that intercalated disc with active gap junctions to move ions from one place to the next. And that leads to irregularities in heart muscle contraction, a loss of normal cardiac rhythm, and then that ultimately is an arrhythmia. So after an MI, a common complication is some type of arrhythmia, oftentimes atrial fibrillation. Our last type of muscle is smooth muscle, and we see here that we have smooth muscle fibers that are one single mononucleated cell, and they can it's one muscle fiber essentially, and they are shorter, elongated, and fusiform shaped cells. So depending on the muscle type, the muscle body, the location, the function, the cell shape can change. It's not always consistent. They also communicate via gap junctions and they're surrounded by endomysium. They're also the only muscle type that's capable of regeneration and mitosis. And they can undergo contraction, which is involuntary using myosin and actin. And they're anchored at dense bodies located in multiple directions. So when they contract, they kind of look almost like this smushed up raisin, for instance. There are no sarcomeres and no striations. So these dense bodies are represented here as this kind of yellowing thing, and they anchor actin filaments. And actin and myosin are not arranged into sarcomeres. They kind of are almost kind of all across the cell. And so when they contract, they pull on these dense bodies and they shorten the they essentially shorten as a whole, which causes almost like a shrinking and ballooning effect to the muscle body. Some other interesting components about smooth muscle, they have caveolae, which are invaginations at the cell's surface that can hold calcium, which can pinch into the cell when they are stimulated. Calcium is also stored in the smooth endoplasmic reticulum or the smooth endoplasmic reticulum. And calcium's claim to fame is that it initiates contraction. Action potentials are propagated via gap junctions, which are located between adjacent muscle cells, and neurotransmitter released are released by the autonomic neurons, or contraction can be stimulated via hormone stimulation. This is showing contraction here or contraction here, depending on if you're using light microscopy or electron microscopy. This is in a relaxed, elongated smooth muscle. You can kind of see almost these like fusiform cell shapes or spindle shell shapes. And this is a cross section of smooth muscle with our nuclei kind of in the center. Smooth muscle is often in a bilayer format. So we can see smooth muscle here. And we can see we have our epithelium, we have our connective tissue base, and then we have our smooth muscle located in between. And those are our three muscle types. Your goal for this week is going to be to get familiar identifying the different structures of each muscle type and to be able to tell the difference between the three in their shape and their structure and their function.